For isolation and analysis, we isolate and purify compounds from plant extract. We use different types of column gels, beads for isolation, some of which are Dione HP20, Cefadex LH20, C18, MCI CHP20P, and these are used with different kinds of column like open column, semi-preparative column, gravity column depending upon the type of gel and amount of sample. First we pack the column with required gel beads using with the first solvent to be used as a mobile phase. After this we load the sample with the first solvent to be used, or we can also go for dry loading if the sample does not get dissolved in the same solvent. We use the fraction collector to collect the eluent in different tubes. After checking the profile of eluent in tubes using TLC, we combine the ones which have same profile. Then we use HPLC to analyze the fraction, that is the eluent collected. Samples are prepared by filtering them in short thread vials where the volume should be more than 500 microliter, and the sample vials are placed in autosampler. We have to make sure that we wash the injector and column before injecting our sample, but at the same time we need to be careful about the bubbles. There should be no bubbles in the syringe. We also need to be very particular about the conditions, which include the oven temperature and the pressure, in order to maintain the consistency in our results. The sample is loaded by injector needle, which takes amount of sample from the vials that have been already fed into the system method according to our requirement. When we have the complete profile of the sample, we go for the purification of the compound using semi-preparative HPLC. This is what system looks like. We inject the sample into the system and collect the desired peak, which is the desired compound. This is how we collect the peak. As the eluent, which is our peak, is very dilute, we concentrate it by using the rotatory evaporator and dissolve it later in the suited solvent. Then we analyze it using HPLC in order to be sure of its purity. From here it is referred to as a pure compound. Once we are sure that it is a pure compound, the next step comes the identification of this unknown compound. The first for which we use UV spectrophotometer to observe the absorbance of the compound at different wavelengths. Then we use NMR 1D for proton and carbon, and 2D where we have two variables, proton-proton and proton-carbon, named as HMBC, which stands for heteronuclear multiple bond correlation, HSQC, heteronuclear single quantum correlation, and COSY, which is correlated spectroscopy. HMBC gives us the correlation between carbon and protons, which are separated by 2, 3, or sometimes 4 bonds. The direct one-bond correlation are suppressed in HMBC, but the information about this direct one-bond correlation is provided by HSQC, which gives the chemical shift correlation map between the directly bonded proton with carbon-13 or nitrogen-15. One more commonly used technique for 2D NMR is COSY. It is a very useful method for determining signals that arise from neighboring protons, which are usually up to 4 bonds. By using the NMR data, we are able to sketch out the structure of the compound, and then mass spectroscopy is used to confirm the mass of the compound. The in vivo model used is female spark dowley rats. They are ovary ectomized. By removing the ovaries, we try to mimic the postmenopausal condition in the women. To achieve this condition, isoflurane is used as an anesthesia to make animals unconscious. Hair covering the abdominal area is removed, and after which ovaries are surgically removed. After 2 weeks, we add either the extract or the compound from the plant orally using the gavage feeding tube. 12 weeks later, rats are euthanized. The blood from the heart is collected in the tubes in order to obtain serum from it, which is further used for toxicity analysis. We separate the femur for 3-point bending in order to observe the strength of the bone. It also provides us information related to maximal load, stiffness, and maximal strength. Micro-CT is used for measuring bone mass density of femur and vertebra, and for the microarchitecture construction of trabeculae. The serum separated from the blood is analyzed in automated hematology analyzer for measuring creatinine for kidney function, aspartate aminotransferase, alanine aminotransferase for liver toxicity. All these are measured to see the toxicity of the extract or the compound on the essential organs that play a role in drug clearance from the body. The in vitro model that we use for osteoporosis is primary human osteoblasts. We maintain the cell at 37 degrees Celsius and 5% CO2. And we have to be careful about the sterile conditions in order to avoid the contamination. The media we use is osteoblast growth media. The doubling time of the cells is 27 hours. This is what cells look like. Cell viability is analyzed by MTT assay. MTT is prepared with the media in the ratio of 1:9. Media is removed from the plates and MTT solution in the media is added to the cells. It is incubated for 4 hours. After 4 hours, MTT is removed and DMSO is added in order to dissolve the crystals. Then we measured the OD at 600 nanometer. ALP assay is done for the activity of alkaline phosphatase. It is the early biomarker of osteoblasts. To perform this assay, media is removed, PBS is added to wash the cells. After this, lysis buffer is added and it is shaken to mix well. Then the lysate is separated for ALP and BCA, which is done for measuring the protein content. BCA is added to the wells designated for BCA and ALP substrate is added to the wells designated for ALP. It is incubated at 37 degrees for 1 hour, and then OD is measured at the wavelengths of 405 and 562 nanometer for ALP and BCA respectively. Mineralization assay is done for assessing bone cell mineralization. To perform this assay, media is removed, PBS is added to wash. (4% paraformaldehyde is used to fix the cells for 20 minutes and then it's removed. And then wash twice using PBS. After this alizarin red stain is added, it is a stain used to determine the presence of the calcific deposition by the cells of osteogenic lineage. It's shaked at 50 rpm for 20 minutes. Stain is removed and double distilled water is used to wash. After this, we take a picture for clear presentation, and this is how picture looks like. 10% cetyl pyridinium chloride is used to destain, after which the red color changes into violet. It's shaked for 30 minutes to dissolve the crystals. Then this violet color solution is transferred to the 96 well plate and OD is measured at 550 nanometer.